Now, welcome back. Okay. So, okay. Let us get back to our PowerPoint now. Shall we start?
Yes.
Yes, okay. Okay. So, this was some processing in the visual cortex. Normally, at our level, graduation level, we don't ask the functioning of visual cortex. What you need to know is there is primary visual cortex, there is secondary visual cortex, and then we have association cortex also. Same like our general sensory system. Primary general sensory cortex, secondary general sensory cortex, and association cortex. Same like that. For all sensation, we have three levels of cortices. Okay, nothing more. This is all you want to remember about visual cortex. Okay, now since we have finished everything, we have seen how the eye, the shape of the eye, the lens, and cornea help in bringing light from the atmosphere toward the receptor. This is what we saw in the first half of the first lecture, isn't it? Then we saw once the light comes in, how it stimulates the receptor. So we saw the receptors are stimulated because of change in retinal protein or retinal chemical, which is a vitamin A derivative, isn't it? So 11 cis retinal is converted into all trans retinal. This simple chain causes a chain reaction that will cause action potential, isn't it? Now remember, the receptors are hyperpolarized by light. Okay, this is one word you have to remember. Once this hyperpolarization occurs, somehow it generates action potential in the ganglion cell, which is transmitted through the optic pathway. You should know the optic pathway as we discussed, and finally it reaches the visual cortex. Okay. In the visual cortex, further processing will occur. So remember, some processing occurs in the retina itself, in all the five cell layers, and some processing will occur in the brain. So this is how we are able to see. Okay, this is how all living beings are able to see. Now, what is remaining is some additional aspects of vision. First is how we are able to see in 3D. We see everything in three dimension, isn't it? So ability to identify three dimension. The third dimension is a depth. So the ability to identify depth is called as stereopsis. So this is the topic of our discussion. First topic of discussion actually related to properties of vision. Okay. So we are able to see the third dimension also because over a period of time, our brain has learned that when the two eyes are bringing information about same object, so it will compare the information of both the eyes. Here you see this eye looks at the object and it looks like this, and this eye looks at this object and it looks like this. You can see the difference in the uh, vision of both the eye. This difference is compared and combined in the brain, and brain has learned to derive depth from this information. Okay, so you require both vision to identify depth. You can identify depth with single eye also. That is called as parallax vision. Forget about the terminology, but it is slightly different mechanism. For our exam, 3D vision or stereopsis or depth perception is because of binocular field of vision. Simple as that. That is all you have to remember. Binocular यानी using both eyes. This is the most common way of identifying depth. Okay, that is shown here also in the picture. You can see if you are watching a 3D movie. Okay, so left eye will see it something different and right eye will see the same picture slightly different. Both these pictures are combined and shown on the television. Okay, so when you wear the colored glasses, the left eye will capture this image, the right eye will capture only this image, and brain will combine it to create 3D vision. This is how 3D televisions also work. Okay. Next thing, this you will do after the lecture whenever you have free time. This is a picture from a textbook called as Neuroscience by Dale Perf. There is a research paper based on this picture. This actually looks like a wall, isn't it? So if you focus on this wall or beyond this wall, what is after behind this wall, focus for a few seconds, you will see 3D objects floating in the air in front of the picture, in front of the picture, okay? Try to look behind the wall for few seconds to minute. You will see 3D structures floating in front. This is how eye is able to see 3D structures. Okay. So you can do this after the lecture and do it. You will actually see a sphere, a pyramid, and a cube in the air. Are you able to see?
Actually, what do we do again, doctor?
Just try to focus your eyes behind the wall. You will be able to see a sphere and a pyramid and a conical shape, not cube. So you have to practice and train your eyes to do that. Okay? Because I have practiced, I can see it within a second. I can see that the picture has got a wall behind, and in front of the wall there are 3 objects which are hanging in air. One is a sphere made of the wall stones, another is a pyramid, and another is a cone shaped structure. Okay. So with practice you will be able to do a 3D picture. This is actually a 3D picture. You need some practice without glasses to see the 3D image. So what I try to do is focus behind the wall, not on the wall, but behind the wall. So after a second or so, I am able to see. And try to move your head, you will be able to, it will help while looking behind the wall. Okay, do this after the exam, please. Oh, I mean, after the lecture.
Okay.
Okay, now we go to second property of the eye, that eye is not only able to detect 3D vision, it is able to detect the colors also. Okay, so let us see how colors are detected. Colors are detected because we have 3 different types of cone. Some cones respond to frequencies which are represented by red color. Some cones respond to electromagnetic wave frequency of green color, and some cones are able to respond to blue colors. Okay, so these are the primary colors. If you know your school terminologies, these 3 colors are called as primary colors. Yani combination of these 3 colors can make any color in the world, isn't it? So they are primary colors and all the others are secondary colors. So we have cones for all these 3 colors. So what happens is whenever light falls on the retina, if the object is blue colored, it will stimulate the blue colored cone 100%. The other cones will not be stimulated. Red 0, green 0, and blue 0. That means the object is blue. The brain will say that only blue colored cones are sending action potential and the object is blue. Okay. Another example is if the cones 83% info action potentials are coming from red cone, 83% from the green cones, and maybe 0 or 5% from the blue cones. The brain will think it is yellow color. Okay. This is how the percentage of stimulation of different cones will tell our brain which color this object is. Yani brain is just trying to imagine based on the percentage stimulation of the receptors. Okay. That is why for some people an objects look different colored, and for some people it looks different colored. You remember that picture of a skirt on WhatsApp? For some it was white and blue, and for some it was gold and silver.
Yes, doctor.
This is how it will. This is how it works. Because our biology is different, the percentage stimulation is slightly different. None of the receptors are made from same machine, isn't it? They are all biologically developed. So slight variations are always there. Therefore, our perception of color is also slightly different. Okay. So, but our point for our exam is color vision is done by three different types of cones. Cones for red, cone for green, and cones for blue color. The percentage stimulation of these cones will determine what color we see. This is called as trichromatic theory of color vision, or also called as Young-Helmholtz theory of color vision. Okay. In our lab, maybe Dr. Amani will take this test. You can identify color vision. Okay. So in general, if you see in this picture, most of us will be able to identify the number 2, isn't it? With this dot. Okay. Yes. For people who are having color problems, some people may miss one cone. One of the three cones may be weak or totally absent. Please mark my word. One of the three cones, R cone, G cone, and B cones, one of that cone may be weak or totally absent. That will cause color blindness for that color and any other color made by using that color. Okay. So there are different terminology. For red color, it is protanope, and then other terms. I don't remember which one is green and which one is blue. But the terminologies are protanope, deuteranope, and tritanope. Okay. This test is called as Ishihara chart. It is present in our lab. Okay. Last part. This is a common we have seen whenever we are walking into a movie theater from our parking, bright light area of the parking. Suddenly, when we enter into the movie theater, when we are going into the theater and going towards our seat, we are kicking everybody's legs, isn't it? And when we sit, and after 10 minutes or 20 minutes, we are able to see their faces clearly and we realize, oh, I kicked that person also, and that person also, isn't it? This is our practical experience. Yeah. Whenever we go from bright light into darken area, we are not able to see clearly. After some time, we are able to see. This is called as dark adaptation. Okay. Similarly, when we are in dark areas and we come out into bright light, for few seconds, everything is very bright and blurred. We are not able to see, and after some time, we are able to see, isn't it? That is called as light adaptation. Okay. So this is dark adaptation and light adaptation. In general, it has to do mainly with the amount of vitamin A and retinol available. Okay. So when you are in bright light, you have used already lot of vitamin A because you are on bright light. Lot of light has entered the eye and it stimulated. So there is not much vitamin A remaining. So when you enter the dark area, it takes some time for vitamin A to convert to retinol and then be utilized by the receptors of the rods. Okay. So on an average, it takes up to 20 minutes for the darkness adaptation to occur, up to 20 minutes. So after 10, 20 minutes, you will be able to see best in the dark area. Okay. Similar explanation can be given for light adaptation also. So this is all. Of our physiology related to vision. The last thing is perception. What brain sees is not about which receptors are stimulated and which pathway is bringing action potential. More than that, it is the imagination of the brain that depends upon our training, background, culture, and our own thoughts. Okay. So if you see in this picture, there are 4 circles, red circles, with 1/4 missing in each circle. Okay. But our training has taught us to think that there is a white square kept over four red squares, isn't it? So this is our perception. Actually, there is no white square at all, isn't it? So that is the difference between sensation and perception. Okay. You want to do this exercise, students? All of you?
Yes, okay, doctor.
Yes.
Yes, doctor.
Okay. So let me tell you about this exercise. You will see a black and white X-ray type of image in the next picture. Okay. So what will happen is your eyes are filled with that black and white image. Yani the receptors for that color are saturated. Okay. This color is saturating you. Focus your eyes on the red dot and keep focusing till this light is there. So your receptors are getting saturated. All the peripheral receptors are getting saturated with the black and white picture. Okay. That causes increased sensitivity of color vision. And that will show you the real color also. Whenever the white screen comes, start blinking. Till then, don't blink, focus. Did you see the color picture?
Ah, yes, we see it, doctor.
Yes, doctor.
Who is this personality?
She's a famous actor.
Indian actress.
Indian.
Indian actress Aishwarya Rai. Okay, she was the Miss Universe in 20 years ago, maybe now. When I was a student at that time. Okay. Okay. You can try this with this picture also. Focus on the red dot. Don't focus anywhere. Just keep focusing on the red dot. So we are saturating your eyes with contrast color actually. That will increase the sensitivity so much that without vision also you will see a picture, colored picture. Keep focusing on the red dot. Did you see or blink? Blink.
Ah, yeah, we saw.
Yes, yes.
Okay.
Benedict.
So this is even though there is no color actually stimulating your retina, but still you are able to see color vision because brain receptors become hypersensitive. Their baseline action potential change going to the brain of certain color because of contrast saturation. So opposite ones were saturated, and that caused the contrasting color or complementary colors to become overly sensitive. So the moment white screen comes, those sensitive ones were sending abnormal or more than normal action potential because they have become sensitive to the brain. And brain thought there is a color picture and you are able to see that, isn't it? So this is how vision works. Many of the things which you see is simply the brain thinking there it is. Okay. So this is all about the vision. Thank you. Who's this personality?
Benedict.
Who's this personality?
This one, I don't know.
Yeah, not Benedict Cumberbatch. I'm not asking about the actor. I'm asking about this personality. He is Ibn al-Haytham. In the Western culture, he is known as Al-Hazin. He was one of the first person to contradict the science of that time. At that time, whatever that century is, the science said that object release, eyes release light on the object and we are able to see. Some sort of energy is released from the eye which will fall on any object and the person is able to see. He was the first person who said it is opposite. Light from the object goes into the eye and we are able to see. And he developed that pinhole camera using the tent. Okay. Also, he is responsible for many of the medicine related concept. So you know he is one of the person from the golden era of Islam. Okay. Scientific knowledge was at its peak at that time. You can watch this video at your free time also. That will also help in explaining this paragraph. Okay. So this is all. If you have any question, please ask.
Thank you, doctor.
Okay.
Thank you, doctor.
Thank you very much. Now I'm going to tell you about the remaining lectures. I think for girls, we are remaining lecture is 18, 19, and then the 20th one. For boys, only 20th is remaining. The 20th lecture is a small one, smell or I think smell it is. Okay, it will take 10 minutes only. So what I am suggesting is the remaining time we will be utilizing for revision for your exam, whatever end block exam is coming, isn't it? So I'll cover I'll utilize the remaining time for revision. So you can keep the 20th lecture online so that we have a revision for all the topics related to your end block. And 18, 19 for girls, I'll take it tomorrow or day after tomorrow, whenever it is scheduled. Usually it takes only 1 hour for 18, 19. It is about hearing. Hearing is a very small concept, not very large. Both the lecture we can finish in the class in 1 hour. So that is all for today. Thank you very much. Let us stop here.